Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes. Of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors given below. Competitive inhibitors occupy the active site and prevent a substrate molecule from binding to the enzyme. Non-competitive inhibitors attach to parts of the enzyme other than the active site to distort the shape of an enzyme.
Uncompetitive enzyme inhibitors definition. Uncompetitive inhibitors are molecules that bind only to the enzyme-substrate complex, S-complex, and not to the free enzyme. This binding usually occurs at a site different from the active site, and it prevents the reaction from proceeding to release the product. Mechanism. 1. Substrate first binds to the enzyme to form the S-complex. 2. The inhibitor then binds to the S-complex, forming an enzyme-substrate inhibitor, ESI, complex. 3. This complex is catalytically inactive, meaning product cannot be formed.